Welcome, everybody. I hope everyone online can hear me. Um, we are trying a new version for this H3D Foundation webinar, where it's a hybrid event. So we do have some people in the room, and we do have some people joining us online from all over the world. So it's a mixed bag, and it's also a double header. We're going to be kicking off with a malaria talk, and then the second talk will be on TB. So firstly, I have the privilege and honor to introduce our first presenter, Dr. Tim Wells. He's a distinguished scientist at MMV, which stands for the Medicines for Malaria Venture. He works with a wide network of partners and scientific advisors to move towards a new generation of innovative molecules, which will help to drive the control and eradication of malaria. Prior to joining MMV in 2007, Dr. Wells was responsible for research at Serono, a Swiss biotechnology company with a large international team. He has worked in industry for 20 years and has also done basic research with Glaxo's Molecular Biology Institute in Geneva. And he completed his PhD in chemistry in London and is an enzymologist by training, which I actually didn't know that about you after all these years. He also obtained a doctorate in biology at Cambridge for his work on cytokine biology. And Dr. Wells is currently a fellow of the Royal Society of Chemistry and the Academy of Medical Sciences in the UK. So with that, he'll be giving a talk on the science of defeating malaria. Is this the correct presentation? Just the wrong title slide. We'll ignore that. Great. So over to you, Tim. Can everybody online hear us? Maybe a thumbs up. Great.
So it's a great pleasure to be here. I have no idea how the hybrid thing works, because for the people at home, then I will have a tendency to sort of walk off.
You carry on.
Walk off. That's why we've got the shower. Okay, it'll catch me. It's good. So maybe a couple of words of explanation. So I work for MMV. So this is an organization that develops and delivers medicines against malaria. As we said, I'm a chemist by training, but this talk is largely aimed at, so you can see from the title slide, it's normally given as part of a Science of Defeating Malaria course, which is run by the University of Ho in Ghana, Dakar, and Harvard. So basically, you can also find different versions of it online, which change year by year as new data comes up. So the reason to give it to this group is think about the course that we're normally teaching is to people from the National Malaria Control Programs. So you guys are all in discovery and early development. These are people who are at the front end of deploying new technologies and thinking about what's coming down the pipeline. And I thought it'd be a useful exercise to try with this group, because quite often what happens in discovery is you're working towards a goal, and somebody old, usually like me, says, actually, do you know what, that's not really what we should be doing. And if you have more of an idea of what happens further down the pipeline, you get some feeling for why we say some of the things we say and how solid they are, right? Because not everything everybody tells you is as solid as everything else. So basically, this is it's probably 45 minutes. We may have to skip some slides, but just to say, talking through what are the problems, what are the medicines we have, both for treatment and for protecting children against malaria, and then how do things develop over the next few years. Okay, so basically I work for MMV. So MMV is Medicines for Malaria Venture, which is a partnership between basically academia and industry, governments and private sector, science and medicine. The goal is to get new medicines out. You can see here we have a team in Geneva. We have a team of about 100. And somewhere you can see where Cape Town is. Yeah. You have
lots of lines coming up.
Yeah, exactly. There's always the thing when you show this is like, who my favorite person is, then you have to be a bit careful. But there we are. So new, safe, effective, and affordable medicines. And so one of the things I hope you get from this is that malaria is a disease where decisions on buying drugs, maybe take it back a step. In Europe, the person who decides what drug you should have is your doctor. That seems wise. He went to medical school or she went to medical school. In the US, what happens is because they have direct-to-patient advertising, quite often it's the patient that goes to the doctor and says, I want that. In Africa, most places, it's the Ministry of Finance that makes those decisions. You'll see it, and it comes up time and time again. Anyway, so there you go. Yes, there's a delay. So this is the portfolio of medicines we have, and it's sort of segmented from the pink stuff that you guys work on, which is discovery. We've got stuff going into preclinical, human volunteers, which is what we call phase 1, through to exploratory studies in patients, so proof of concept, combination studies, regulatory, and new products. If you're interested in this slide, then it's actually on our website, and the one on our website is much more useful because you can click on the slides and you get a one-slide summary of what's going on. So if you ever want to know what's in the pipeline, it's an amazing resource, and we update it at the moment every year, so it's usually pretty up to date. So 3 things today, quickly, just how do you prevent children getting malaria? Because prevention is better than treatment. Then what do we do to treat them? And then what sort of tools do we have for eradication? So if you give a talk on malaria drugs, what's really interesting is that nobody ever talks about vector control. If you give a talk about vaccines, it's interesting nobody talks about drugs. So for this course, let's say protecting children, you have vector control, you have drugs, you have vaccines. So let's take them in that order. So vector control, what's really exciting, if you say what's been exciting in the last 2 years in malaria, is that the WHO has now recommended new sorts of bed nets. So these are a combination, one of them has basically something that boosts the effect of the pyrethroids and overcomes resistance, and the other is a small molecule growth regulator. What's interesting about these two, just to take home, is that up until about 2010, most of the protection of children was done by vector control, not by drugs. So that's well documented. Beginning of the century, then the bed nets stopped working, and that takes time. It's not overnight. These bed nets would have been really useful if they'd been available in 2015. And so one of the questions, and it'll come up again in the drugs, is to say, well, the insecticides used there were deployed in the United States in the late 90s. So it's taken us 25 years to gather the data to get WHO, we as a community, 25 years to change policy. So that's a question for the future is, how do we make the whole system more efficient? And then the other thing for later, hold on, there is an innovation premium. So these are more expensive than the old nets. And then the question for the countries is that worth paying for? And we'll come back to that at the end. So I work on medicines. Probably the most exciting thing in protecting children from our perspective is this thing, seasonal malaria chemoprevention. So in the Sahel, which is that blue bit of Africa, so just south of Sahara, there are just over 50 million children under the age of 5. You have a season of malaria, which is between 4 and 6 months. And so what the Malaria Consortium started to do from 10 years ago was give every child in the Sahel every month a course of SP, sulfadoxine pyrimethamine, plus amidaquine. Compliance is about 90-something%. You say, why? Because what happens is they distribute the drug, and when they come back the next month, the mothers know that it works because it has 80% efficacy. So they see the effect. So that tells you something that when you're taking a new drug into Africa, decision makers are community leaders, the imams, the church leaders, and the mothers in this case. So 80% efficacy or 75% efficacy in the rainy season, currently being used on 53 million kids. Cost of this, and that was where we came in. We worked with the manufacturers on the costings. We managed to get Fosun to commit originally at about 16 cents. So it's now 20 cents. So that means protecting a child for a year is about $3.69. In Nigeria, where they have lots more children to protect, the numbers are lower. Right? The more children you have, the cheaper it is. And there's a discussion at the moment. So 2 things. One is this is currently being manufactured by 2 partners in Nigeria, one in Kenya, one in Cote d'Ivoire. And the question is, can you get your African manufacturing partners up to international standards? And with a product like this, it is actually relatively straightforward. It's just quite a lot of paperwork. Yeah, I think this is worth saying. So on the course then, on the course we had Nena, who's the National Malaria Control Program head in Nigeria. So Nigeria, country, 200 million, so it's got 40 million small children. In the north it's seasonal, in the south it's not seasonal. You say they're basically, you can see how this program has rolled out over the last 10 years. And now pretty much everybody in Nigeria takes the SMC. And if you read the paper, it's really interesting because they talk about how you have to work with the community leaders and how you have to influence people. So it's not just a question of, I bring the drug and it will take off. Does anybody work on resistance here? Who works on resistance? So, interesting thing about SP, of course, is that we know that it doesn't work because we have DHFR, DHPS resistant markers. And so you would go to meetings for years and years and say, well, this can't possibly work because we have SP resistance. And I think that's a call out to people to say, we have to be very careful to distinguish between clinical resistance, in other words, drug failure, and seeing markers. So this is a really good example. So in Mozambique, so next door to you guys, they have seasonal malaria. SP amidiquine was not deployed until 2022 because we knew it wouldn't work. The experts there said, no point doing it, it won't work. Finally, Malaria Consortium in 2022 managed to raise money to do studies in Ghana, where it also shouldn't work, and in Mozambique. And basically, it works fine. Why does it work fine? Well, basically, you've got 3 drugs in there. So the question, how much weight is the amidiquine pulling? How much weight is the SP? But the outcome is 90, well, that one is 80, 86%. So it's better than the average in the rest of Africa. And again, it's cheap. So for those of you who haven't seen the ways that on the right-hand graph, it's the with and without SP amidiquine, how many cases of malaria you get. So it's pretty dramatic. SAS drugs. So we've done drugs are $3.90 or whatever. They involve mothers having to give drugs once a month through the season. Mothers actually quite like that. The other alternatives, we could have a vaccine. So you have to start by saying, you know, we're all used to the idea of yellow fever or mumps or whatever vaccine. So vaccines against protozoa, like malaria, there aren't any, or there weren't any until we had that. So the first thing is to say, people give the idea or get the idea that making a vaccine against malaria was easy. It's not easy. The parasite has ways of evading the immune system, from changing its surface antigens to controlling the immune system and so on and so on. So it's remarkable that any of this works at all. And it's a tribute to the fact that people work for 30 years on basically turning up and up and up and up on the adjuvant in terms of the strength. So we have 2 vaccines. One is from Glaxo. So the originator vaccine was from Glaxo. It's called RTSS. It has the CSP1 antigen, so the bit that sticks on the sporozoite, which, do we have TB people here?
Yes.
Yes, good. So it's when the sporozoite, so the bit that comes from the insect, goes into the liver cell, that's the first point of contact. So basically, they have a whole pile of that stuck on a hepatitis core antigen. You can see that's the yellow bit around the side. So it's not the world's best malaria vaccine. It's actually quite a good vaccine against hepatitis B. So then Oxford came along 20 years later. Instead of using Ceravecii, they used Bishop Asturis. They had a better expression system. They have exactly the same adjuvant in there, pretty much. I mean, it has a different name, but it's based on the same saponins. And the price difference is really interesting because GSK is the first out the gate. Somebody says, how much do you want for this vaccine? They say, wow, $10. Nobody really knows how much it should cost because the principal driver for the cost is not the epitope, it's the adjuvant. And the problem that everybody has, whether it's Glaxo or whether it's Serum Institute, is it's the same adjuvant that you're using in a commercial vaccine. So something like herpes virus or HPV or whatever, where you can charge $200 a dose. And there's a limited supply in the world at the moment of the saponins. So anybody who's young and enterprising, set up a saponin company and yeah. Okay, so what about vaccines and drugs together? So there's a couple of papers in the New England Journal, so that's probably the wrong reference. But looking at this, in somewhere like Mali, child gets basically 1 productive infection every year. It's a good number. So it's 1,100 infections per 1,000 children. On that, you can see if you give a vaccine or you give SMC, you drop that number from 1,100 to 300. So that's the 75% efficacy, whether it's drug or SMC. If you give them both together, you get another drop. So that was really exciting because there were lots of people who said, yeah, but you know, amidiquine is a really good immunosuppressant. Maybe this wouldn't work. So actually, if you had enough money and enough time, you could actually reduce malaria by tenfold tomorrow. The question is, it's actually really complicated to deliver. So the interesting thing here is that they're following it up for 5 years, and without going through the paper, what you can see is the SMC works fine for 5 years, probably for 7 years now. The question is, will the vaccine still be adding at the same amount in 5 years' time? The only reason to say that is quite often people miss that if you want to have 5-year protection, your study takes more than 5 years. Right? So the TB crowd, when you say the M72 vaccine has taken people 12 years to develop, and you say, well, that's because you're looking for 3 years protection. So the longer the protection, the longer the studies take, the slower development. This is the next one's from Asragani's group, so Hillary. So don't worry too much about the right-hand graph, but they produce new versions of this table with every paper they write. But just to give you an idea, cost per disability adjusted life year. So you've done disability adjusted life years. So basically you look at how many years of life are lost, or how many years of life you consider are partially lost. So feeling handicapped or losing your sight or something counts as 50% lost and so on. So they have tables to calculate this. In our case, it's mainly death. It's the death of a child. You lose a 5-year-old, that counts as 50 years of life lost. So nets for people that don't have them. If you go to Central Africa, then, people fish with them, people play soccer with them. They don't protect you from mosquitoes if you use them wrongly. SMC, $24 at current prices. Moving to the new, more expensive nets, $32. So WHO has guidance that these numbers, it's actually quite good value for money if these numbers are lower than the per person GDP in a country. So in a country like Mali, that would be $900. Here it's what, 5,000, 6,000. So these are really low numbers. Seasonal vaccination with the expensive vaccine is 155. Seasonal vaccination with the cheap vaccine is probably 70. So you start to say, compared with other interventions, these are still dirt cheap. And so one of the questions now is, you've got all these interventions. How do you convince people that they want to spend their healthcare budget on this rather than something else? The right-hand graph, I have never really understood, but you can read it at your leisure, but the thing to take home is there's a massive inflection point at around $1. So if you understand nothing from the graph, it says that when they're modeling it, the vaccine becomes really efficacious and really useful from a public health point of view when it gets down to $1. And so in a meeting, anything you can think of that might bring the vaccine down to $1 would be really good progress. And then just finally, the better vaccines, then what we're looking for, everybody would like a long duration vaccine. So this is one slide summary of immunology. Remember, these vaccines are given 3 times, which means they're given at 14, 18, 22 weeks. It means the child is protected from maybe week 30. That's when the clinical study starts. And then they're not indicated for children beyond the age of 3. So what would be nice is that you had a vaccine that you give once, like yellow fever, and you get 3-year protection or 5-year protection or whatever. We have lots of ideas how to do that, right? And there's stuff in here about, B-cell responses and so on. But just to say, it's a fiendishly difficult question, right? Because the B cells go to sleep, the T cell, or the B cells forget about the antigen. The T cells actually show T cell fatigue markers. So the parasite is doing some very, very clever stuff to tell the immune system, don't worry, which we don't see in viruses so much outside of HIV. So basically, yeah, could we get a virus with, sorry, could we get a vaccine with a long response? And the answer is, we hope so, but we're right at the beginning of understanding this. One of the things that's really nice, we do studies with volunteer challenges. I don't know if you've seen some of this. So we take Australian volunteers, we infect them with malaria, and then we cure them. The beauty of that from an immunological point of view is that we can take large sample volumes of blood. So we can take a couple of mils of blood from a volunteer, which you can't take from a child. And so actually the understanding, if you look at some of the technology in immunology now, next 5 years is going to be a massive wave of interesting human immunology. And those of you who work on mouse immunology will know that the usual problem is you learn something in the mice and then you go into man and it's completely different. So that's the reason to be optimistic. What else can we do to protect? So if you look at the little chap on the right, then he doesn't really care about B-cell immunology. He cares about 2 things. One is, how much does it hurt? And it has to hurt a little bit, otherwise people bizarrely don't think it's a very good vaccine drug, but not too much. And then does it stop me getting malaria? So we had a couple of things. One is, could you do a monoclonal antibody? And the other is, could you have a long-acting injectable? So again, thinking about small children, thinking about me, injection volume should be about 0.5 ml. That would be nice. 1 ml, not too much more than that. So we had an expert meeting in 2016 where we did a target product profile. And that's actually referenced there. But basically saying that we would want an injectable that would do the same as SMC, so 80% protection over the season, preferably from one injection. Everybody here is bright and young, so we don't need to explain monoclonal antibodies, but say that the beauty now, of course, is that when I was young, then we used to clone antibodies and humanize them, all the rest of it, whereas now we can clone them directly from the patients, or we can have mice that have complete human immune systems. So basically, we get good antibodies straight off the bat. Why is that important? Well, it's important because I'm
dramatic pause.
Yeah, dramatic pause. It's important because okay, so if you think about monoclonal antibody, then we've got 1 ml of injection. So the number, if you remember nothing else from the talk, the monoclonal antibodies are soluble 100 mg per ml, sometimes 125. So the fact that we use numbers like 125 says you never get much above 150. So take 100. So if you can get 1 ml into somebody's muscle, you can get 100 milligrams. So the first thing is everybody says monoclonals are so expensive we'll never be able to use them. You say yes, but if you can make 100 milligrams cheap enough, right? So the numbers are there's quite a tight band. So when we started this, and we were working with the guys at NIH trying to build the business case for this. So antibodies, nobody will tell you this, but it's in the literature. If you manufacture them at scale, cost around $50 a gram. Hopefully from this talk, you'll learn that in my job, then actually knowing how much things cost is quite important. We can scale, you don't need to know that. Allometric scaling in biologicals is a piece of cake. Yeah, that's the other thing. And they have special bits on the constant chain where you can change the half-life of the antibody. So it gets recirculated and pumped out. But basically, yeah, if you have an antibody and you can get it into a 1 ml injection, that injection probably costs around $5. It's a starting point. Remember, the vaccines are $3, so $5's a bit expensive. If you do, this is really nice review on on antibody targets, monoclonal antibody targets. So the only thing you need to take home from this is they go all the way around the life cycle, and there are a lot of them, right? So just add up in your mind how many different genes there are there. One's in development, well, there are 6. 5 if you discount one that's actually against it's great. One that's against the host target on CD147. So why the discrepancy? Why there's so many good ideas and so few projects? And the answer is that you can prove an antibody works quite well in a cell biology assay. You can do an animal study with it, but the moment you talk about going into humans, you need a GMP batch. And the current price of a GMP batch is $12 million. So you're trying to raise, as an academic you're trying to raise $12 million based on a mouse experiment. Small molecules, then the preclinical batch costs us maybe 50,000, right? So this is an interesting one. Technically, the only people who can make monoclonals in the academic sector are universities who have their own pilot plants. And that's why you see basically Radboud and Oxford pretty much are dominating, and the US NIH, obviously. Good. So that's the front runner antibody at the moment is this one. It's not quite good enough. So go look through the papers later. But basically, you can see nice tail that you're getting exposure out to 8 weeks in a phase 1 study. And in a phase 2 study, so this was published last year, you can see you get this sort of 70% protection over 24 weeks. So single injection will protect children for 24 weeks, 70%. But you look carefully, we're giving you a 300 milligram injection, so that'll be 3 mils for an adult. So it's a bit high and it's a bit expensive, but it's saying it's on the edge. So this team, based on our discussions, this team will now look at how can we protect really small children, because the really small children have the advantage that they're really small. So getting 30 mg per kg into a child that size is much easier than one that size. Okay, so you guys, some of you working on long-acting injectables. We talked about this over lunch. Yeah, okay. So send some slides on this, but the original long-acting injectables project was done by Pfizer, or Park Davis, that became Pfizer in the 1960s. So they did a study where they made antifolate drugs, so cycloguanol palmitoate, an insoluble salt, in an oil injection. It never happened now. And they injected and they protected children for 6 months. So the first question is, it's perfectly possible. It was an awful formulation. If you read the paper, they talk about, the heroic people who tested it, because in terms of joint, basically pain at the site of infection, it's injection. But it was possible. The problem is that because it's an antifolate and it was a single drug, then when children failed, then they had drug-resistant malaria in them. So you really do have to pay attention to what the emerging infections are going to be. Second thing, so when we first started talking about this, everybody said, it's going to be hard. WHO approved cabotegravir. So if you want to learn anything about innovation in malaria, we steal ideas from other therapeutic areas. And that's why I come to TB meetings. But HIV is so much further ahead. So HIV, take cabotegravir, it's a 2-month injection. And within malaria, we said, well, 2 months isn't possible. We don't have those half-life. And then suddenly cabotegravir comes out and you say, well, actually, you don't need super duper long half-lives. You need to flip-flop the kinetics so that the drug doesn't leave the injection site, but it's doable. What you also know now is we have lenacapavir for HIV. So FDA is talking about either 6 months or 12 months protection from a single injection. Those of you who work on LAIs, just go back and Google the 2 structures for cabotegravir and lenacapavir, and you'll see that one of them is awful, and that's the lenacapavir. So it's designed as an injectable. So probably for us, first generation things, we're taking drugs that we know that'll give us a couple of months protection, but if we really want a year's protection, we have to design things. I don't think I've got any data in here, but this is our program. So we have basically a version of atovaquone. So those of you who take Malarone, then this is basically an acetate of atovaquone at the top. And then there's another electron transport inhibitor, so another mitochondrial transport inhibitor for a different site, which comes from an American group. The nice thing about these two is they're actually synergistic because they work on the same pathway. So in vitro, then 1 + 1 gives you 4. And they have similar PKs. So the top one is currently in a phase 1 study where it's doing fine. And you know it's doing fine because I wouldn't talk about it if it wasn't. So when people give confidential talks about things where they can't present the data, then the fact it's on this slide means it's looking good. So the summary, and this is in Miriam's paper, so we did a review of target product profiles for protection, is to say, well, oral standard of care, it's there now, 53 million kids, it's cheap, it's manufactured in Africa, has everything going for it, but it's complicated, and it will eventually fail. Vaccines, yeah, it's a bit heavy, it's a 50% response, and it's this whole thing of how do you communicate 50, 70% responses. It's still quite expensive, but that should come down with a bit of competition. The monoclonals, they will protect everybody. So as the vaccines just protect children between 6 months and 36 months, in theory, the monoclonals could protect anybody, and the long-acting injectable is the same. The difference between the monoclonal and the long-acting injectable is probably that we can do the long-acting injectable 10 to 100-fold cheaper. We'll see. Okay, so then that's preventing. How long do we have? We started 5 minutes late. Yeah? She's ruthless. I don't know whether you... We'll see. Just yawn at the back if you're... Okay, so we're going to flip to... Yeah, it's going to go back to what? Here we go. So how do we treat patients? Artemisinin combination therapies. So it's artemisinin, which is a Chinese natural product, with a partner drug, and the partner drugs are derivatives of quinine through chloroquine. So the 4 aminoquinolines, amino alcohols. Always 2 drugs, always the same artesunate. Currently, there are 5 combinations approved by the agencies, by WHO. The nice thing across the top, nice packaging, got all the... So the slide right at the beginning I didn't show is say, I am not endorsing that you buy any of these, just to be clear. There are generic versions of each of them, and the generics are actually really important because generics are actually what drives the price down. It gets competition in. So, treatment shortening, once a day versus twice a day. One of them is twice a day. artemether-lumefantrine. 4 of them are once a day. So logically, the market should go to the ones which are once a day and not twice a day, because that's what we teach. You know, we tell you guys to come up with treatment shortening, less complexity. So you see straight away who makes the decisions. The person who's buying it. So this is just for the public sector. This is not in the private market. In the private markets, then people are buying pyronaridine and DHA piperacin for themselves. So what do we have? These drugs originally are all tested in adults. They're then tested in small children, very small children. So the big breakthrough over the last 24 months is that we had enough data, and I'll show that on the next slide, to show safety in first trimester pregnancy. This is really big. You know, when we explain, I'm going to do something totally inappropriate and say, is anybody pregnant here? But you're not allowed to do that anymore. But it's a really interesting question, because when you talk about pregnancy, everybody says, yeah, but you know, we can detect pregnancy. And you say, no, the embryonic growth in the first 8 to 9 weeks of life is when they're fragile. That's when you get all the birth defects. Even in our family, then knowing whether my wife was pregnant in the first 7 weeks is quite tough. And you say, so the problem is, if you don't know they're safe in first trimester, then is it really a good idea to be giving it to women at all? And so this is a major breakthrough. It was always used in adult women, and there were, quote unquote, never any problems. Now we know that there were no problems, or at least no worse problems in the background. So one of the things we're doing is looking at first trimester pregnancy. And what's been interesting about that, so without going into it, what the team were doing in Liverpool and in Kenya, so Stephanie Delacour, 3 lines down, was that they had digitized the antenatal clinic data, and then they digitized the malaria data, and then they compared the database across. So for the first time, just being able to digitize those 2 databases, you could tell everybody who is pregnant and gets malaria in the first trimester and is treated. So this was a breakthrough, because most parts of the world, you can report an adverse event or you can report your pregnancy. Central Africa, that doesn't happen. So out of 40,000 women that went through that study, we had 40 inadvertent exposures in first trimester pregnancy. So people who took the drug and didn't realize they were pregnant. That's enough, right? So once you've got 40 and you know that there's no major problems, then the ethics people are then quite happy for us to say, well, we could deliberately expose people in the first trimester. It's going to be safe. So it's not going to be a major issue. Now we need to understand the subtleties of it. So that's what we're currently doing underneath this SAPPHIRE study supported by the European Union. But what's nice is it's a way of getting drugs tested in early pregnancy, early on in the programs. The artemether-lumefantrine, the time between approval of the drug and this first trimester recommendation was 24 years. So what we're trying to do is basically say, once we've got an approved drug, how quite fast can we show that it works for everybody? Second one is Novartis. So the dogma was that children under 5 kilos, so children in the first 2 months of life, do not get malaria because they're protected by the antibodies in the breast milk. The pediatricians then say, well, if that's the case, then why do I have children that are getting malaria, newborns? Some of it is genetic malaria transmitted from the mother, and some of it is just exposure. But basically, they'd be doing a study on a new ratio, right? Because as you go down in weight, then the different metabolism changes. So it turned out that there's not enough lumefantrine in the children to treat the children who are small. You can't give them double dose of artemether-lumefantrine because then you hit the toxicity for the artemether. So basically, they went back and did a study in newborns, and there are like 30 of them in this study where they're looking at the PK. So you can imagine the complexity of it. And that's now been rolled through, and it's with Swissmedic, it's with the WHO, and it went to Ghana first as a regulator. I should say, you know, we're now dealing with African regulators who are one level, so they're called maturity level 3. So whereas Swissmedic is maturity level 4, the African agencies, there are 7 of them, want to be at ML4, like South Africa, by the end of the decade. And this is really interesting because you see here that Ghana as an ML3 is saying this is fine. And people in Geneva are saying, well, who are you to say it's fine? And they say, well, we're the people with the patients. So this is going to be a big change. And I think that's where South Africa will play a key role because it's one of the leading regulatory authorities. Quickly on artesunate resistance. So It first comes up in severe malaria. So severe malaria, the patient comes in, they're unconscious, and they have a lot of parasites. So the trick is to get rid of the parasites as fast as possible. What basically they saw in Southeast Asia at the beginning of this, so this is 2009, is that the half-life of the parasites, so that's not the drug, is going from about 3 or 4 hours to 6 or 7 hours. And that basically was the first sign of a problem. As far as the patients are concerned, then actually they come down later. The clinical impact, yeah, they have to stay in hospital a day later, 2 days later. But so far, even after 15 years, we're not clear whether that has any detrimental effect on the long-term survival of the child. Two things. One is we've never seen a super slow parasite. So when it first came up, we said, well, the half-life is 6 hours. Imagine it goes to 26 hours. Then the patient's going to be in the intensive care unit for 10 days. This is not good. And eventually it gets to the point when we're not killing them any faster than they're growing. We've not seen an IC50 shift. And that's important, or an MIC shift. That's important because the toxicity for artesunate kicks in at about 3-fold higher dose. So we know that from overdosing people. And so to say those two things, if we ever saw a super slow parasite or a shift in the IC50, we'd have a major problem. We have Kelch mutations, which is the mutation that causes this. They had them in Southeast Asia. Now we have them in Africa. And the phenotype is exactly that. The parasite is cleared slower. What's interesting is that WHO's guidance, if you see these slow clearing parasites and you're not happy as a clinician, then you should give quinine on top. So there is a solution. It's not the end of the world. So this is my quick teaching slide on the kill kinetics. But say on the left-hand panel, you see artesunate in wild type and mutants. You see the half-life of the parasites is much better in the wild type than the mutants. But even in the mutants, it's still only 7 hours. Quinine historically, so that's data from the 1950s, but quinine historically has about the same sort of kill rate. But quinine is an awful drug because of its side effects. Right? So quinine has chinchonerism, it works on the cardiac sodium channels. It causes hypoglycemia. So you give it to the patient may go into a hypoglycemic coma, which is a bit of a problem because they also have a malaria coma. And so historically, one of the problems of quinine is that the doctor has to stay with the patient because you have to keep an eye on the sugar levels. With artesunate, they didn't have that problem. So the major driving thing for treating physicians or treating nurses is you give artesunate, you can go home. You give quinine, you have to stay. So there's a little bit here of saying, well, even with the Kelch mutation, the speed of cure is still the same speed as quinine, right? It's not. Then the final, some of you know, Cipagarmin K609 from Novartis kills like crazy, right? So half-life is, so the parasites are all gone in 8 hours. And so the interesting question will be, there's no doubt, so this is currently in a phase 2 study, so it's in children that have severe malaria. There's absolutely no doubt that it'll kill the parasite faster. The question is, how will it change the journey of the patient? So is it good to kill so fast? I think so, but we'll see from the data. WHO says, though, that it's actually the partner drug which is driving resistance. And if that's the only thing I show you, then that's really important. So ACTs, the artesunate slows down the cure, so they still have parasites on the 4th day, but they always get better. And we measure better by how they're doing at day 28. If the ACTs fail, it's because you have resistance to amidiaquine, ironardine, all the partner drugs. And they came up in Southeast Asia, and they've been seen sporadically in bits of Africa. So one of the questions is, how do we best protect the partner drugs? So if you go to a malaria meeting, everybody's talking about artesunate and what we do. And you say, no, the best thing we can do is protect the partner drugs. So what can we do for that? Got sort of 4 boxes. You've seen the severe malaria now. The first thing is always, what can we do with what we have? Right? Sort of drug discovery is great, but it takes a long time and it's quite complicated. So what can we do better? So actually, I showed you those 5 ACTs. The last one to be registered and approved by the WHO was in 2022, right? So although we tend to present it as that was what was happening 10 years ago, the last decade, 2010 to 2020, had been finishing off the paperwork and finishing off the trials for all the different ACTs. Number 1. Number 2, multiple first-line therapies. So it is truly bizarre that the world uses the cheapest ACT, so artemethyl so much. You know, it's okay. You know, you could understand half of the world's supply was artemethyl, but the idea that some countries only had one drug because it's too complicated to have two, and then you worry about resistance. So what WHO has been trying to push towards is say, well, countries should have more than one option available, and when they worry about one, they should stop using it and use the other. What you saw in West Africa was we had amidacin resistance 15 years ago in 5 countries. It's not there anymore because they don't use amidacin. So the rotation through, it's like crop rotation. You know, it's always going to work. The other thing we're working on with the Chinese is why don't you put, instead of rotating, you just put all the drugs in the same tablet. So that's more a TB approach. Not all the drugs are working perfectly, so put them all together and it should be fine. So there, then it works quite nicely because you can give it twice a day, so the amidacin dose is split, and so from a safety point of view, it's nice. And the other thing that's important is that triple artemethyl lumefantrine amidacin will be roughly the same price as the current drugs. And remembering that all of this is driven by who buys the drugs, then when we come out with a new drug, we just have to be a bit careful that it's not too expensive. Briefly, and that was in my portfolio slide at the beginning, but this, you see, we're not yet into discovery space. Yeah, so in phase 2 at the moment, so phase 2 is basically going from 15-year-olds with malaria all the way down to 6 months children with malaria. We have 2 combinations. Carbamaquin is the Merck Dundee compound. Zydus is the AstraZeneca compound. They're currently looking at phase 2 studies on a single dose cure. Why was that important to the companies? They say, well, we've seen all the stuff in front of us is 3-day cures. That there are at least 7 or 8 drugs which are 3-day cures. You know, if we're going to create a market, we have to do something better. So treatment shortening is really important there. Ganaplacide lumefantrine is the lead Novartis product there. So that's KF156. That's currently finishing a phase 3 trial this summer. So the nice thing is that if we did have a catastrophic artesunate resistance, you actually have a combination that we could use. And then, and I skipped through the slides because we're running short of time, the question is, well, what if we get lumefantrine resistance? And everybody said, well, it's very difficult to get that. Now we have some lumefantrine resistance, but again, we have to see how that plays out. Good. So the questions are all for the class. So I'll leave the deck with you, but with the class then that is supposed to afterwards, we spend an hour discussing things. It's really interesting because it's all about, if you look at them, it's all about, if you're in charge, how do you do prioritization for public health policy? So this is what we're looking for. So this is now back to H3D, and what does the next molecule look like? So this is probably the last slide that's useful to you. Probably the only slide that's useful to you. What are we looking for? Well, we need something that's fast acting. And as you saw then, you know, pretty much most things we work on when we get into humans kill the parasite over 3 days. There's very little that we do that's slow these days. So that's pretty much a no-go, that if your drug is really slow killing, then there's so much that we have that's better. Refractory to resistance. So this is a big debate, right? Discovery team will say, yeah, 10 to the 7 is fine. Cipagarmin has a resistance threshold of 1 in 10 to the 7. In phase 2, they see breakthrough resistance with monotherapy. Now, they argue, as we do in TB and malaria, that what we're going to do is going to put it in combination with something that doesn't have, you know, that basically kills everything. But it's still a bit worrying because it's saying that 2/3 of the patients that fail have resistance. So if your combination cures 100.0%, you'll be fine. But if it only cures 99.7, then of that 0.3 at the end, then so I don't know. There's a bit of, you know, we internally say, well, I think we can cope if things have an AMR of 8, but be careful because that number will change. And that sometimes we talk about, you know, MAV shifts the goalposts. You say we don't shift the goalposts. The drugs in front of you shift the goalposts. We just comment on it. You saw a low treatment dose. I don't know if I've got it in this slide, but we've seen over the years that the development compounds have a human dose that's gone down from 1 gram to 400 milligrams, sometimes to 100 milligrams. And that's really important because we're giving 2 drugs and all the gubbins that goes with it. And then secondly, you know, drugs cost a certain amount to make. And if you want to do something cheap, the easiest way to do it cheaply is to get the dose down. So it's much easier to work on a low dose drug to get it cheap. No food effect, because children that have malaria can't eat food. They vomit. Children that don't have malaria, when they're finishing the course of treatment, are hungry. And so any drug where you say, well, don't have some food at the beginning and do at the end, it's too much. So anything, food effects are a real problem for us clinically. And then ideally, when I was saying, half the population is female, obviously, but of those, there are quite a lot of people in that sort of, if I was in Europe, I'd say 14 to 45, but in the places we work, then, the sort of 10 to 45 group, so the complicated sociology, but the chance that a girl becomes pregnant in some of the areas where they have high transmission is much higher than around here. Anyway, so keep it clean for pregnancy. Yeah, you've seen all these. This is how we do drug discovery. For the TB people, exactly the same as the way you guys do it. We have very clever people that came up with assays, some of them here, to screen parasites against large collections. We come up with hits. Then across this group and others, we come up with a network for testing in different assays to get a fingerprint of how things work. And then we set up collaborations to then optimize those compounds and move them forward. This is what we've done over the last 12 years. So this is quite important for the malaria team. We'll get there. So these are, this is the year, so it's 2006, 2022. This is the predicted human dose. We have some quite good ways of predicting dose. And you can see that it's coming down. So we produced a new TPP in 2016, 17, and the world responded to it. Right, so this is us moving the goalposts, moving the goalposts. You can see here that when we do resistance on the other side, then this is the MIR, so 6, 7, 8, 9. I put on this 7.5. You can see that basically all the compounds have gone into the top right-hand corner, which is where they should be. And you can say, if you're this project, you should be feeling a bit exposed. So that's what
Whose project is that?
you can work it out. I'm not saying live on camera, but it says a little bit to the teams. Then, if you don't know what's going on in everybody else's project, if you don't know how the portfolio is going, then you will not see the changes in the TPP happening. Because obviously this line is totally arbitrary, right? We move it up and down. So we put it somewhere where we can actually capture most of the compounds. This line then, 400 milligrams in an adult is quite a lot. We used to have people who say, well, 2 grams is fine. And you say, well, try it. Okay, do we have 2 minutes at the end? No,
I think
definitely not. I think we'll stop there then. That's good.
Two parts.
Thank you.
Because I'm sure there are questions. Do you want to end off with a dramatic ending?
No, I'd just like to thank you all. No, but seriously, it's one of the joys of working with Cape Town is you have a tremendous sense of energy. You have lots of people who are committed to working on malaria. I've just sat through a TB meeting where everybody seemed to have my level of grey hair. So it's nice to be back with young people who are actually doing drug discovery. It's a long journey. So that if you saw those graphs of, what the global malaria community have managed to develop in terms of drugs over the last 15 years, it's pretty impressive. And you played a role in that. It's the same for TB. I don't know why I think you guys work on TB.
No.
It's the same for TB, right? It's one compound every year. But the problem for H3D is that your next compound may be 7 or 8 years in the future. It may be tomorrow, and it's very stochastic. And so when you're trying to center your emotions on how are we doing as a group, how is the field doing, you have to look at how the entire field you're part of is working. Otherwise, you just go crazy. But say, this is, an absolutely amazing place, and it's amazing because of you guys.
Yeah.
So that's my end on.
That's a great.
Thank you.
Everybody's doing and questions. I see we already have one in the chat. Yeah. So do you want to read it out, and then we'll go to warm them up, and then we'll ask the room.
Oh, that's a very long chat. Bryce says, great talk, Tim. I was asking if ACT failure might also be due to artemisinin-induced dormant or quiescent forms. In fact, when treated by a patient with ACT, artemisinin, which is a fast-acting drug, rapidly exerts its activity, inducing the formation of dormant parasites.
Good question.
By that time, the partner drug will find no parasite to kill after 48 to 72 hours.
Yeah. So basically, the question is, when you look at the kill kinetics of parasites, then we can detect 10 parasites per microliter by microscopy. We can detect 10 parasites per microliter by PCR. So there's a threshold where you can't see the parasite anymore, even by PCR, because you can't take enough blood. Right? So if you take 10 mils of blood, then your limit is 1 parasite per 10 mils. So the question is, when we give a drug like I showed you the Novartis frontrunner for severe malaria, when we give a dose of 10 milligrams, then we can calculate that that's enough to kill 10 to the 20 parasites. And we don't get a sterile cure. So if you look at, they come back later. And so there are a couple of questions, for all drugs, and it's particularly true also for artemisinin. Is it that there's some compartment of drugs somewhere, they live in the lymph nodes, I don't know where they live. There's a compartment of parasites that we're not getting to just because they're in a different place. Or is it that it's a life cycle stage that the drug isn't working at, so they're quiescent? But either way, then what's very clear is that so far we don't have any new drugs which are sterilizing. Right? The drugs keep the parasite away because there's a plasma concentration of drug. So that's going to be interesting. As the new drugs come forward, then that will be one of the deep questions is, do you get all the parasites and how do you get all the parasites out? Because if you don't, then the patient comes back to the clinic. Some of the patients come back after 42 days or 63 days. So yeah, it's a good question. It's exactly that. That could be one of the reasons that we're just not getting to the parasites.
Questions from the room?
Total stunned silence.
Stunned. I was curious to know what Pan Africa is. That I hadn't heard of that.
You're very observant. When you work with the European Union, well, first of all, everything has to have a name. So the PAM Africa is a clinical consortium of clinical trials that's supported by an EU grant. Do not ask. I'm the lead investigator, but don't ask me what the PAM bit stands for. So it'll be something anti-malarials, progressing anti-malarials for clinical trials in Africa. But you just pick the words you want. Yeah. So it's a collaboration. I mean, that's why I was in Mozambique. So it's a collaboration between us, Merck, Novartis, Lamborghini, which is with Tübingen. So Lamborghini is in Gabon with Tübingen, and for reasons that we can explain over coffee, the site in Manisa, Mozambique, is tied up with Barcelona School of Public Health, and that basically we have 3 clinical programs that have been going forward. So one of them was the Artemis lumefantrine under 5 kilos. But yeah, great. So we're currently renewing that grant. So. We'll see.
Yes, we also have an EU grant where we came up with a fun name. We landed up with Rafiki.
Yeah, exactly.
So yeah, exactly. Yeah. And then another question online is: do multiple infections lead to development of immunity, which can protect from new infections?
Exactly. So, if you get yellow fever, nobody here. Don't even ask that question. If you get yellow fever or no, the disease. So you get yellow fever is a bit like getting Ebola or getting whatever. Then you have a sterilizing infection. Sorry, sterilizing immunity. So people who get viral infections like that, if they die, that's because their immune system hasn't coped. If the immune system copes, then they've built up a sterilizing response, so they never get the disease again. So that's why in an Ebola outbreak, the survivors then can become nurses that look after the rest. With malaria, then, the problem is that you don't get a sterilizing immune response. So you get it once, you get it twice. So the theory is you probably get it 50 times as a child. So you see maybe 10 clinical infections over the first 10 years, but they've had it lots of subclinical times. And that's what builds up this semi-immune response. So the question is, if you could do something, if we understood enough of immunology to say, instead of a child have to be infected 50 times to get a semi-immune protection, could you do something that they only had to be infected 5 times? And so I think that's a sign of, that's that's what we need to be working on understanding the immune system. And there's a whole talk about that. But the problem is that you get malaria once, you're not protected. And it's quite short duration. So what we see in Europe is very high infection rates with people who go to Africa. So Africans going to Africa from Europe because they know they can't get infected. So the returning travelers tend to be people who are born in Africa who've lost their immune response. And you ask them, they didn't take drugs because they don't need them. So yeah, multiple infections. So if we understand that one, then that would be a big breakthrough to understanding how to make a decent vaccine.
Awesome. Thank you, everybody, and thank you for the engagement online.